Okay, so if you are absent on Monday, December 6th or Tuesday, December 7th, you're missing some really important notes that it's going to be hard to understand unless you watch this video and you listen and not only copy your notes but understand the process. So it's a little like math class. You have to know how to do it. It's not just memorizing vocab. So last week after our DNA test, we compared DNA to RNA. And we are looking at a process that's really important in biology. And we are on this page of our notes packet. So we're still on the same notes packet we've been doing. So you need to find that. Push pause if you need to, and you're filling in your notes. So here is the central dogma. The most important thing about biology is understanding that DNA contains the inherited information used to make proteins. So that's the process we're going to go through today. There are two steps to making proteins. Transcription, where DNA is used to make RNA, and translation, where the RNA is decoded into amino acids to make proteins. So in order to do transcription and translation, you have to understand that RNA is a little different than DNA. Three big reasons is RNA sugar is called ribose, and RNA is single-stranded instead of a double helix, and RNA contains uracil, there's U's instead of T's. So when we do make RNA in transcription, it's going to look a little different, and Chargaff's rule is going to be a little different. So when we make RNA, we are doing transcription. It's the making of RNA. So your body's going to get signals called promoters when you need protein. When you need protein, your body's going to tell you in your cell to make RNA first. That's the first step. So transcription is the first step. In bacteria, it happens in their cytoplasm because they don't have a nucleus. So that's a good review at this point before finals. No nucleus. In a eukaryote, this is all happening in your nucleus because you do have your DNA inside the nucleus. So how do we make RNA? Well, just like with replication, you need enzymes to speed up the process. So RNA polymerase is an enzyme that speeds up the making of RNA, so it speeds up transcription. So remember, DNA can make DNA, that's replication. DNA can make RNA, that is transcription. So transcription, it uses only one side of the DNA as a template because RNA is single-stranded. So what that means, if we look at let me see if I can make that a little bit larger. If we look at our notes, if I have a strand of DNA and I know the letters in DNA, I'm going to use Chargaff's rule. T is still going to go with A, C still goes with G, G still goes with C. However, A matches with U when we're making RNA. That's the difference. So if I tell you to do transcription, in transcription, all I need to do is my matching letters. So just imagine your body gets a trigger, that promoter signal tells your body to make RNA, and that's called transcription. So to make RNA, it's going to say, okay, I've got a T, that means I'm going to make an A, C, G, A, U. And you are just writing the matching letters. That's all you have to do. What we're going to learn is these letters code, and in the second step in translation, we are going to look these up on what we call a codon wheel in order to find out what they mean. So step 1, make RNA, transcription. So in our second example, C goes with, G goes with, and you're just doing your matching letters and trying not to mutate. You want to make sure you get the correct matching letters. So I'm going to go back to my notes slideshow, but you're going to flip to the next side of your notes. So when we talk about making our protein, we are going to do the first step, which is transcription, making RNA. Then we're going to do a second step where we translate the RNA into amino acids, because remember, amino acids are the building block of proteins. And those proteins make your trait. So what we're going to be doing is either making your eyelashes, your freckles, your hair, all of the parts of your body. And that's why it's called gene expression. We express our genes by transcribing and translating our DNA. And the genetic code is universal. All organisms go through a process of it, and that process depends on 3 letters at a time. 3 letters at a time. So we're going to look and practice this 3 letters at a time. And if you were in class, I was giving you this codon wheel. And this codon wheel is how we figure out how to translate the 3 letters. So you're going to start in the center and work your way out. These are all amino acids. They're names of amino acids, and amino acids are the building block of protein. So if my first DNA letters is GGT, I have to transcribe them, then translate them. So GGT, if I want to make RNA, I have to put the matching letters. So GGT goes with CCA. When I want to translate, we can't do it all automatically. Your body is doing this automatically. We need to look it up on a codon wheel. So if my first code is CCA, I start in the middle at this first C, then I'm stuck in the C corner, and I go out from that. So my first code was CCA. That means CCA translates to proline. So my first step, make RNA. My second step, translate that RNA into an amino acid. So let's say my second DNA is CCA. CCA transcribes to GGU. Then I have to find my codon wheel. And by the way, this is on It's Learning in the announcements. And GGU means glycine. And you can abbreviate and just write the first 3 letters. DNA, CGT. Think for a second. What are my matching letters? And then those matching letters, that RNA letters, that's what I look up. So I'm looking up GCA. So again, I'm in the G corner, C, then A, alanine. So this codon wheel will be on your final exam. Last practice, TAC. Transcribe it into RNA using the matching letters. Look it up on the wheel. Methionine. So that's the basic process you need to know how to do. That's why I said it's kind of like math class. But let's make sure we understand how it's happening in your body as well. So when we look at our body, you've got this whole chromosome that is made up of your DNA, and you have sections of your DNA which are your genes. And those genes make, when they're expressed by transcription and translation, make up all these amino acids that are joined together to make a certain protein. So the codon wheel, again, we start in the middle, we go out to the next level, out to the next level. Everything about your gene expression is based on 3 letters at a time. So if I had ATA-GCA, it would be UAU, CGU is my first step. That's transcription. And then I can break it into groups of 3 and look up the amino acids. So, in order to picture how it happens in your body, you have to know more about the 3 kinds of RNA. So the 3 kinds of RNA do the job that we just did on paper. The first kind is called messenger RNA, and we abbreviate it as mRNA. Messenger RNA is what we are making during transcription. It is the message that is carried out of the nucleus into the cytoplasm. So remember we talked about your DNA is stuck in your nucleus? The messenger RNA is what we're making that carries the code out of the cytoplasm. And remember that every 3 is an important number. We call that a codon, which is why we use a codon wheel. So the mRNA is what we look up on the codon wheel. Once the mRNA gets out into the cytoplasm, it's going to find the ribosomal RNA, which we abbreviate rRNA. The rRNA makes up the ribosome. So the mRNA comes out, it attaches to the ribosome, and remember, that's a review. Ribosomes are where we make protein. So the message has to get to the ribosome because that's where your body makes protein. Then we need a 3rd kind of RNA. Transfer RNA, which is abbreviated tRNA, transfers each amino acid to the strand. So its job is to bring in the correct amino acid based on the codon. And it does that because it has a matching code. And if you look, if this is my codon, AAA, the tRNA has an anticodon on it that matches. So we call the code on the tRNA the anticodon. It goes up against it. And what your body is going to do is form bonds between each of those amino acids. And a polypeptide chain, also known as a protein, is made. And it'll keep making it and adding and adding and adding and adding until it gets to a stop codon. So our next step is to practice this. Okay? So I'm going to enlarge my paper because you're going to have to be able to do this on a final exam. So first of all, you've got 2 steps. You have a transcription where we make the mRNA, and then you have to look up on the codon wheel to do the second step where we translate into amino acids. This step in the middle is what your body does. We don't actually need that step to do transcription and translation. But I want you to know this step because this is what's happening inside your cells. So first of all, to make your mRNA, you have to do transcription, just like we've been doing. Match your letters. So I've got an A, G. Then I would break this into groups of 3. Remember, a codon translates on the codon wheel every 3 letters of the message. We're decoding the message. And we're going to put that in this box. So AAG, I would have to look up AAG, and that would be my amino acid. And then I've got CUC, Then I have GGU. Then I have ACC. So these are my amino acids that are translated, decoded from my codons of transcription. So there's step 1, step 2. So in your body, because your body doesn't have a codon wheel, scientists figured out the codon wheel. The way it knows to bring in lysine is because the transfer RNA anticodon matches the mRNA. So to do the anticodon, this is the order I do things in. I go from codon to translation, then I go back and I fill in my anticodon. Anticodon is the opposite. If it's AAG, my tRNA is going to say UUC. CUC, GAG. GGU, CCA. ACC, UGG. Notice, transfer RNA is still RNA. You have U's, not T's. The only place you have T's is in your DNA. Okay, so in class we are doing this and then flipping over and doing the next page. So you should do all of that because on Wednesday and Thursday we're going to review and talk about mutations, and then this will turn in this DNA notes packet. So make sure that you have caught up. I will be in study tables Thursday after school if you need one-on-one help with this.